Hey guys, and welcome back. You should have already watched your Unit 5 content video for the day on Tragedy of the Commons. Also, we went a little bit over clear cutting, deforestation, things like that. Today we're also going to go over experimental design because, again, we know for sure you're going to have an FRQ on it. It's going to be the one where you're probably going to get the most points. And it's probably going to be the one where it's the easiest too, because if we understand everything that we need to have in experimental design and you have it and it's reasonable, you can get a big chunk of your points for your AP test there. Okay. So this is a reminder of what to include on your FRQ for your experimental design. I would say go ahead and just pause this and study this. Make sure you know what you need on the FRQ before you get started. So you're going to describe your expected outcomes. You're going to state your hypothesis, and that hypothesis must be testable. You're going to make sure your design is possible and consistent with what's required. So what's required, what they ask of you in the actual question. Specify the independent, the dependent, the constants, and the control. We're going to practice that today. Describe how you're going to collect, graph, and analyze the data. So how would you present this data? Okay. Even averaging data could be considered analysis. You guys averaged your data when you did your air pollution experiments. State how you'll make conclusions. And then you always indicate how you will repeat the experiment. So you have to verify it, right? So state that more trials will be conducted, and you can give a number. Say greater than 50 is good. So say this trial will be completed 60 more times to verify results, right? You add that in the end, and you show the readers that you know what you're talking about and you know what would happen in an actual lab setting, okay? So like I said, pause this video, make sure you understand what is required of you. Okay, so let's just review what the different components of this question actually are. Remember, this is scientific method. You guys have seen this since your freshman year. It's review. I'm confident that all of you guys know how to do it, but we're just going to get some refreshers just in case. So remember, your independent variable is what the scientist changes from group to group. So often it's just whatever the experimental variable is. So the way that I always remember it is independent variable is the thing that I change. So it's what you are manipulating about the experiment. Your dependent variable is the condition that results from the change in independent variable or what the scientist measures, okay? So your dependent variable changes in response to your independent. So your dependent variable is dependent upon your independent variable. So if you can just remember it's what you measure, the dependent variable is what you measure, you can find it every time because you go to your scenario, or if you're writing your scenario, you have to make up what you're going to measure. But if you're doing an example and you're reading a scenario, whatever the scientist or the student is measuring, whatever they're copying, the results that they're taking, that's your dependent variable. Your control is the normal condition or the standard for comparison of the other experimental groups. You have to have a control. If you don't have a control, your experiment means nothing because you have to have a baseline to compare it to. You have to be able to look at it and say, oh, this is what it's like under normal conditions. And you have to have that because you have to be able to show that your manipulation of whatever variable it is, whatever you're manipulating actually has an effect on the dependent variable. So you've got to have that control or else your results mean nothing. And then you've got your constants, which are experimental factors that stay the same from group to group. Now, constants are not the same as your control. Your control is what you're comparing everything to. Your constants are what you try to keep the same throughout your experiment because you never want to test more than one variable. Okay, that's important. That is important when you guys go to write your FRQs. You can only test one variable at a time because if you test multiple variables, you don't know which one is making the effect, if that makes sense. So like if you wanted to measure the effect of water and sunlight on a plant, on plant growth, you would need to do trials just measuring sunlight. And you would need to do trials just measuring water. You can't do them at the same time because if you were varying your water and your sunlight and you see a difference in the growth, well, how do you know it's it's both of them and not water or sunlight. You know, does that make sense? So a good experiment has multiple constants, right? And it also tests one variable. And then your hypothesis, basically it's just a prediction about what's going to happen. And remember, you write it as an if-then statement. So if I manipulate this, aka my independent variable, then this will happen. This is what's going to happen to the dependent variable. Okay. So let's take a look at an example really quick before you guys do this on your own. So it says, Anne read that there had been a sewage spill in one of the local creeks that runs into the local river that's used for recreation, electric power, and drinking water. We know all that. She also read that the factor that causes fish kills in the water is the lowering of dissolved oxygen. We've also learned about dissolved oxygen, right? She learned that the microorganisms in the sewage were using up the oxygen, but if the weather remained cool, the dissolved oxygen level would probably remain acceptable. She collected samples of water from the creek, took them back to the lab, and placed them in several containers of the same size. She put the same amount of water into each container. She varied the temperature of the containers from below room temperature to above room temperature, but kept each at a constant temperature. So that means that whatever temperature she chose, so below room or above room temperature, she just kept them at that temperature each day. So like if the below room temperature was, let's say 30 degrees Celsius, or not 30 degrees Celsius, 30 degrees Fahrenheit, 30 degrees Fahrenheit, then like the next day, she would make sure that container was also 30 degrees Fahrenheit. She would keep it at that constant temperature. Each day she performed a dissolved oxygen test on the samples and recorded the data. At the end of the 5 days, she was able to draw conclusions from her experiment. Now, I'm about to go over everything that you'll need in your FRQ and identify it in this with you, but if you want to pause the video and try to identify your independent variable, dependent variable, control, and constants, go ahead and do that, but I'm about to go over them. Okay, so let's start with your independent variable. So remember, your independent variable is the thing that I change, or the thing that the scientist changes. Here, Anne is collecting these water samples. She wants to see what the effect of temperature is on dissolved oxygen. So the thing that she is manipulating here is going to be the temperature. Okay, so temperature is going to be our independent variable because she is varying what the temperature is from day to day, or she's varying the temperature in the different containers each day. So our independent variable is our temperature. Now let's find our dependent variable. So dependent, remember, is what you measure. It's what changes in response to the independent variable. So let's see if we can read and figure out what she's measuring. So it says, each day she performed a DO test on the samples and recorded the data. There it is. She's recording and measuring the dissolved oxygen. So your dependent variable in this case is the dissolved oxygen. The amount of dissolved oxygen is dependent on the temperature. Do we see how that happens? So we're measuring the dissolved oxygen and it's changing in relation to the changes in the temperature. So temperature is independent, dissolved oxygen is dependent. Our control. Now here it doesn't explicitly state our control, but remember that our control needs to be the normal conditions, right? So we have something to compare our data to. So here, because we're varying below and above room temperature, our control would just be a container of the creek water that's at room temperature, okay? And I'm assuming maybe room temperature is what the temperature is outside, like in the creek, like the environment that she collected this data from, but it's what is what normal conditions are. So for this, the normal conditions would be like the temperature that's outside. So we're going to assume it's room temperature because it doesn't explicitly say, but like your constant here would be the water at room temperature, because that's what you would compare your results to. And then the constants. So remember, constants are the same that are across all variables. So there's a few constants here. We've got where the water comes from, right? The water comes from the same place for all setups. The amount of water is kept consistent. The size of the containers is kept consistent. And then it's consistent in that she holds the same temperature across the days as well. So all of those are constants. They're not your controls. They're not what you're comparing it to, but they are constant in the experiment. And then hypothesis. So let's think of a good hypothesis. So remember, hypothesis is an if-then statement. So if I manipulate this, then this will happen, okay? And in scientific writing, you don't really want to say I. So you would state it more like, so for this particular example, we could say, if the temperature is lowered, then dissolved oxygen will increase. That would be an acceptable hypothesis. So you don't say if I lower the temperature, you just say if the temperature is lowered. Because again, in scientific writing, you don't necessarily want to have personal pronouns. You want to keep it kind of analytical. Also, another thing for this, so if you didn't want to specifically say lower temperatures versus higher temperatures, you could kind of do like a blank statement across the board. You could say, if temperature is manipulated, temperature and dissolved oxygen levels will have an inverse relationship. And now me saying that they'll have an inverse relationship, this tells the reader that if I increase temperatures of the water, dissolved oxygen will decrease, right? If I decrease the temperature of the water, my dissolved oxygen will increase. Those have an inverse relationship. So that's a little bit more like scientific. But if you're not comfortable wording it that way and you think that that'll trip you up, just do it the simple way. Remember, you guys aren't going to get points for having the fanciest writing. You're just going to get points for it being correct. So stick with what you know. Stick with what you feel comfortable with. I think this is a review for everyone. You guys, at the beginning, we did this, a review of this early on when we kind of touched on scientific method in our very first 2 weeks of school. So if you have any questions, let me know. Your assignment for the day are going to be scenarios like this. There's a Google Doc in the assignment. If you open it, it will ask you if you want to make a copy. Go ahead and make a copy. And it'll have, I think there's four or five more of these scenarios, and it just asks you to identify the independent, dependent variable, constants, give the hypothesis, and that's all you're going to do. So go ahead and do that. You can either download it as a Word doc and upload it as a file. I have it set to where you can do a file upload, or you can share it with me, and I've included my email in the assignment as well. So if you guys have any questions, let me know. Keep on working, keep on reviewing. We are almost there, okay? I hope you guys have a great day, and I hope that you are all doing very well with this continued quarantine. I will talk to you guys soon.